TALENs stand for transcription activator like effector nuclease. Like zinc finger nuclease, TALENs are also artificial restriction enzymes used in genome editing. They are modular in form and function and are generated by fusing a TAL DNA binding domain to the cleavage domain of FOC1 restriction endonuclease. So TALENs are composed of two domains. One is a TAL DNA binding domain and the other is the cleavage domain of FOC1 restriction endonuclease. These two domains are fused to form a monomer of TALEN. TAL DNA binding domains are derived from TAL proteins. TAL proteins are bacterial effectors. They are secreted by some beta and gamma proteobacteria. The cleavage domain of TALENs is derived from FOC1 restriction endonuclease. The cleavage domain cuts the DNA within a spacer sequence. Like zinc finger nuclease, TALENs also function as dimers. Dimerization of TALEN proteins is mediated by the FOC1 cleavage domain, which cuts the DNA within a 12 to 19 base pair spacer sequence that separate the two flanking TAL DNA domain. This figure shows the dimerization of TALENs. This is a molecule of double-stranded DNA. Here there are two monomers of TALENs. Each monomer consists of a TAL DNA binding domain and a cleavage domain of FOC1 restriction endonuclease. The TAL domain binds with a specific sequence of DNA. Two such monomers assemble to form a dimer which cuts the DNA within a spacer sequence. TALs are typically assembled to recognize between 12 to 19 base pairs of DNA. Each TAL DNA binding domain consists of a series of repeat domains. Each repeat is approximately 34 amino acid residues in length. Unlike zinc fingers, which recognize DNA triplets, each TAL repeat recognizes a single base pair via its amino acid residue at position 12 and 13. These amino acid residues are known as repeat variable diresidues. The amino acid residues that are most commonly used as repeat variable diresidues in order to assemble synthetic TAL arrays are Ni for adenine, Hd for cytosine, Ng for thymine, and Nn or Hn for guanine, where N stands for asparagine, I for isoleucine, H for histidine, D for aspartic acid, and G for glycine. TAL DNA binding domains can be assembled by a variety of methods. The most straightforward approach is Golden Gate assembly. Compared to zinc finger nucleases, TALENs offer two distinct advantages for genome editing. First one is the time and experience needed to assemble a functioning nuclease is relatively less. Second one is TAL nucleases exhibit higher specificity and reduced toxicity. However, TALENs are substantially larger than zinc finger nuclease. They have a highly repetitive structure, so the delivery of TALENs into the target cells remain challenging. So friends, this is all about TALENs. For more updates, please subscribe my YouTube channel, Sam Biology Class.